Hi there, everybody. Good morning to you. Glorious day here in London today. Warm welcome to everybody for our 6th annual ISNTD Festival, where we're going to be exploring various SciComm initiatives, perspectives, and the impact of those to alleviating disease burden, ranging from disease awareness all the way through to kind of concrete campaigns, bringing that lived experience of the people through. The running theme for this session, for today's festival, this year's festival, is really about citizen science and that kind of extra level of engagement needed to reach out into the various communities affected by NTDs and global health issues. We've had a first session this morning where we looked at inclusive SciComm, and really the gist of that was rethinking the images, the narratives that kind of are trying to paint, as it were, disease and the community per se. So looking at ways of rethinking that. That was the first session. It's brought us neatly into the second session, public engagement and citizen science, from disease awareness to data. This is very much a forward-looking session. So we're very kind of honoured to be joined today by the Natural History Museum in London, Dr. Aidan Emery, Dr. Tine Huyse, and Dr. Mercy Ashepet from the Royal Museum for Central Africa, the ATRAP project, Dr. Mags Leighton from the NTD Network office at Durham University, Professor Marco Salvemini from the University of Naples in Italy, and Professor Matt Todd from University College London School of Pharmacy and his very interesting open mycetoma project. I think we're going to kick the session off straight away because it ends at 12:15, so we've got a good hour and a half to get stuck into everything. I want to hand over the floor to Dr. Aidan Emery from the Natural History Museum here in London to talk about a few issues around SciComm for them. I'm going to push you over to Aidan. Aidan, could I hand over the floor to you?
Okay, thanks very much, Cameron. So I'm just going to prefix what I'm going to say with a bit of disclaimer that I'm giving this talk very much from the perspective of a scientist. I'm a biologist, and I work on NTDs, mostly on schistosomiasis, who happens to work in a museum environment. So, and the reason that this disclaimer actually has a certain amount of significance because the Natural History Museum in London is a very large and complex organisation with a number of different objectives and layers of complexity really within its organisation. So I just want to go through some of the things that people think we are. So firstly, are we a visitor attraction? Well, yes, the Natural History Museum is very much a visitor attraction. The Associating of Leading Visitor Attractions in the UK just released its figures for 2021 just last week. And the Natural History Museum is scored highest with 1.5 million visitors. And that's in spite of a lot of disruption through COVID. And that makes it the most popular indoor attraction in the UK. Is it an archive? Well, yes, it is. And we should probably consider the collection part of the museum to be really what defines the museum as a museum. We've got over 80 million items in the collection, and I worked out that for every painting in the National Gallery, which they've got about 2,500 paintings in the collection, we've got 30,000 objects in our museum. So you could, and unlike paintings being more or less flat and stored and maintained in the same way, there's a lot of complexity and diversity of format and value. Are we a research centre? Yes, we're a research centre. We've got about 300 scientists working at the museum, and we publish about 700 scientific papers every year. And are we an educational establishment? Yes, the museum provides learning resources and supports visits from schools. And we train about 100 postgraduate students from various universities. So it's nice to be able to give out these figures, and we're very proud of them. But for the context of our perspective on NTDs, we should consider the benefits and tensions of working in an environment with such diverse objectives, and where we kind of fit into it. So what we've got is a massive collection, the product of generations of sampling from the world as an essential reference resource. Now, we're doing our best at the museum as an organisation to improve access as much as possible, and a key part of that is to digitise it. So two aspects of digitisation. One is just so that people can see what we have in the collection, which means a publicly available database of all our specimens, and a lot of progress has been made on that over the last decade, and you can see a link to our data portal there. So here, in this case, I've done a search on our data portal for phlebotomus. That's the sandfly, one of the sandfly genera that transmits leishmaniasis. And so if I select one particular sample, this one is phlebotomus perniciosus, which is one of the principal vectors of visceral leishmaniasis in the Mediterranean. And then this is also a SYN type, which means it's one of the specimens that was used to originally describe that species. And then we can even then, it's variable, but for some of the collection, now we've got it fully digitized, and that we've got images associated with it. And here we've got the male genitalia of phlebotomus perniciosus, and that's not just because entomologists are a bit weird. The male genitalia are one of the key diagnostics for identifying the different species. So this is a way of making things very much more accessible. And for these, particularly for the slide material, that's pretty much giving you as good access as that you would get by visiting the collection and seeing what we have. It works very well, of course, for these kinds of samples. It's a bit more complicated, say, for pinned insects or parasites in jars, that kind of thing. But so in some cases, there would be it would be a more complicated process to go through to digitize them. One of the things that researchers who visit the collection are very interested in doing, particularly over the past 20 years, is genetic studies. And for that, of course, you need to sample from a specimen, extract its DNA and sequence it. And not only is that not necessarily a great use of the collection in that they've not been stored with DNA in mind, but also the curators are very keen that we don't hack the specimens to bits, because future generations may wish to access those as well. So the museum has now started to archive material also with that kind of destructing sampling in mind. And so here's some pictures here of the molecular collections facility here at the museum, which is basically a biobank where we store samples in the best possible environments for, particularly for DNA extractions, and also with that destructive sampling in mind. I got involved in this kind of biobanking project myself with funding from the Wellcome Trust. They funded us for 10 years for our to make available our schistosome collections that we'd brought together through decades of working on schistosomiasis. So that's a legacy of material that has come to the museum in the past, plus new collections through projects and partnerships that we've been working with over the past few years. So just as an example of how you can add value to samples by doing that, here's a paper that was published based on some samples collected in West Africa in 2007. And then another paper here from a separate study, in partnership with the countries involved, and then another one from East Africa in 2012. Each of those studies produced its own publication, and then we maintained those samples in the biobank. And then a collaborator came to us. This is Tim Anderson, who works at the in Texas. He had some Brazilian samples, and he actually published a paper on those as well, which I haven't included here. So that's an additional one. And then by bringing samples and data together from each of those, it's possible then to add value to the whole thing by doing a completely separate and unique study, in this case on oxalinequin resistance genes and their origin in New World and Old World populations. And of course that would be beyond the scope of any individual project to generate those samples. Lots of issues, of course, with doing this kind of work. Funding is, and continuity is a problem. The Wellcome Trust we're absolutely brilliant in funding us for 10 years, but of course they can't do it forever. It means we can maintain the collection within the museum, but we can't do the same work to expand and support people in the field doing additional collecting. We have to, of course, respect the ethics and the permissions of the original collections, and since the Nagoya Protocol to the Convention on Biological Diversity came in, we have to comply with the access and benefit sharing legislation of the country of origin. That is, we absolutely fully support access and benefit sharing, but navigating it with the resources that we have can be problematic. So it just makes us less nimble on how we can approach this. The museum, of course, as stated, it's a visitor attraction, so we have lots of opportunities to do engagement. So here's an example of a nature live talk. I know that Cameron has been involved in one of these with us one time. Various formats, but it's usually an interview and a Q&A moderated by a member of the engagement staff, and we do that to visitors and school groups, and we've got facilities to do that in the Attenborough studio. So that's an additional thing that's come in really over the last 10 to 15 years that the museum has started to do to get its scientists up front and out there. So this is an example of one session that we did on disease, particularly neglected tropical diseases. We can use our spaces to host events. So we've got images here from Parasite Day that we did a few years ago. It's very relevant to what we're talking about here. And top right there, that's European Researchers Night, where we get all the scientists up front to talk about what we're doing and really spread the word on our topics of interest. The museum also is really expanding its digital output. So we've got 2.3 million followers on Twitter, so a big following there. Many of those Nature Live talks are on our YouTube channel, and of course our website is really the place where we can do a lot of engagement and get our personal message across. But what we have to think about is where we fit into the picture as people working in neglected tropical diseases. There's a lot of competition. We have a relatively small number of people at the museum who are working on NTD related topics. We don't have any permanent exhibition at the museum that's aligned to NTDs. And as non-engagement staff, we have limited opportunity to provide input into those public facing activities. So the bottom line, really, the closing statement is the same as the opening statement, which we are a large, complex organization, which is interested in many topics. So it's hard to get the attention and maintain momentum, even as people working there to get the message out for our particular field of interest. I should say, just as an aside and just as finishing off, I've been talking to Anna Georgeson of the SCI Foundation recently about the possibilities for an exhibition, and we have found it difficult to get momentum going within the museum. So and it's it's easy to understand why it's difficult for us to get attention when there's so much out there for the NHM to cover. So if anybody is interested in working with us on this, then get in touch, because probably the more people that we can get involved, the more lobbying that we can do within the museum. So I'll stop sharing. So thanks very much. That's me finished, Cameron.
Thank you very, very much, Aidan, for that. Very, very interesting insight into the kind of the very important role of the museum moving forward. I mean, you mentioned the work there with the resistance genes, and how that could not have happened without those samples.
Yeah.
And I wonder how this new realm, this new horizon of digitization is going to play out when you've got things such as virtual biobanks being set up by Find, as an example. We had a session with them earlier in the year around that. So I'm pretty sure there's a rosy future, but it might take some realignment of remits and kind of boosting some of these silos that you're talking about in terms of education remit, research remit, archive remit. I'm sure that's going to come up in the questions in the Q&A, but I'd like to thank you very much, Aidan, for that.
Thank you.
Much appreciated. That takes us to our second the two speakers we've got in second for this session, Dr. Tine Huyse and Dr. Mercy Ashepet from the Royal Museum for Central Africa in Belgium for their ATRAP project. We had a session earlier, a few months ago, I think it was just before the new year started, with Dr. Tine Huyse and Mercy and the team, the full ATRAP team. That's on our website. And it's kind of a new evolution for museums, real engagement. There's something to build on in terms of Aidan's presentation. So I think enough from me. I'm going to hand the floor over to Dr. Tine Huyse and Dr. Mercy Ashepet. Just actually before I do that, a quick shout out to some of the wonderful the participants. We've got from South Sudan, Dr. Bill Garang. Hi there. We've also got from Emery, Dr. Jimmy Kihara, Dr. Abbas Khan from Pakistan, Anna Fyron from Germany. We've got, you mentioned Dr. Anna Georgeson from the SCI Foundation. Anna's there. Hi, Anna. Sarah Mohammed from South Sudan, from Nigeria, Edith Nwanko, and from Tunisia, from the Institute Pasteur, the head of lab, Dr. Ikram Guizani. So thank you very much. And please do feel free to kind of put your questions in for this, for the thing. I'm going to back out. It's enough from me, and over to you, Tine and Mercy.
Thank you, Cameron. Hello, everyone, and welcome to this segment where we shall be talking about citizen science and snail-borne diseases in Africa. My name is Mercy, and I'm a PhD student at KU Leuven, but also at the Royal Museum for Central Africa. I'm very privileged to be co-presenting with Tine Huyse, who is my supervisor. So we'll start with citizen science. What is it? It has several definitions, but typically it is a practice of public engagement or participation in scientific research. As depicted by this image here, we have participants in Uganda collecting data on snail-borne diseases. That is coming later. If we look at types of citizen science, it comes in very many shapes and forms, but most of them are classified or categorized based on a level of participant engagement. And at the bottom, we see contributory citizen science projects, where most projects actually fall. These are expert-driven and mainly focused on collecting large-scale data. Then we also have collaborative citizen science projects, which are also expert-driven, but the participants can contribute in various stages of the scientific research, for instance, also data analysis or data dissemination. And at the top, we have co-created citizen science projects, which are volunteer or participant-driven, and they are focused mainly on finding solutions for challenges in the society. When we ask who is using citizen science, quickly looking at this map of number of citizen science publications per country, prepared by one of my other supervisors, Jacobs, we will definitely see that there is a lot of citizen science work going on in the global north, in the Americas, parts of Europe, and very, 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 very few examples exist in Africa. And yet, if we contrast this with vector-borne diseases, we see that most of them continue to prevail in Africa, and yet this is where citizen science is not occurring. So there is a huge untapped potential for citizen science in Africa for the control of vector-borne diseases. We made a case for this in our paper, a recent paper. You can find more information there. But quickly, you might be asking why citizen science for vector-borne disease control, and there are so many reasons. The WHO has set ambitious targets to be achieved by 2025 to lower the burden of vector-borne diseases. But there are several limitations attached to this, to the current control strategies. For instance, vector control requires a large amount of information or data, which is kind of unachievable because it typically relies on experts, on trained specialists, and these are few in Africa. It's also constrained by resources, by time, by funding. There is a very low temporal resolution in terms of the places where these vector-borne diseases occur are actually remote, hard-to-reach areas. Also, the current strategies are implemented in a top-down fashion, most times kind of not engaging or not widely engaging the local community members. And this is why in our paper we make a case for citizen science, and there are several reasons, or there are several benefits attached to citizen science. I'll focus on three. The first one is boosting data collection, engaging citizens or local community members in vector surveillance or monitoring provides what we call many scientific eyes, and this could complement expert observation, expert monitoring, and therefore provide the much needed data on vector ecology and distribution. Citizen science also allows a bi-directional flow of information from the citizens or from the public to the scientists, and therefore it does not only enlarge our knowledge, but it also, allows us to learn from each other, both the scientists and the local communities. And lastly, citizen science increases ownership by engaging, including the participants, the local people, from the very low stages of the project. It includes ownership and can boost uptake of control strategies. And this is where we now go to snail-borne diseases. And Tina, you take over.
Thank you, Mercy. Yes, so here we will provide an example. And in this case, it's schistosomiasis or bilharzia. It's a disease transmitted by snails, and it affects over 200 million people worldwide. Symptoms include liver and bladder fibrosis, infertility, and it's important to know that a good treatment is available, but no vaccine. So each time when the people are entering contaminated water, they will be reinfected again. So this is why we want to focus on prevention. And this can include snail control, and it can include behavior change. Because if people will stop urinating in the water or defecating in the water, then eggs will not be transmitted anymore to the water and the snails and so on. But to achieve snail control and this behavioral change, we want to call upon citizens. So in the ATRAP project, we involved 25 citizens in Uganda, 25 in Congo, and they are helping in monitoring the snail distribution every week at a specific set of sites in order to create infection risk maps that will guide targeted snail control. But at the same time, they will be involved in community outreach to increase awareness and to induce behavior change. So how do we do that? In the first part, where the citizens will act as snail collectors, they are equipped with protective gear, with a snail scoop, and with a mobile phone on which the Kobo Toolbox app is installed. So they will collect the snails, they will count and sort them according to the genus. They take a picture. This will be uploaded using Kobo Toolbox onto a central server, which can be accessed by ATRAP PhD students who will perform remotely a semi-automated data validation. And then after this, they will provide personalized feedback back to the citizens. So this feedback has been shown to be really important because you see here on the graph that the number of errors which are indicated in orange significantly reduce after feedback. So we had the first feedback in 2020, and then you also see right before the refresher training, the number of errors were increasing again, and after the training, it went down again. But to really look at the scientific quality of the data, we compare the data of the citizens with data gathered by so-called experts, which are ATRAP PhD students, and they go to the same sites and they collect the snails. They will look whether the snails are infected by means of shedding and PCR. And that data and the citizens' science data will be used for spatiotemporal modeling and forecasting of snail populations under different climate scenarios. So if we look at the data we have so far and we compare the citizen collected data versus the expert data, we see for Bamphalaria species in terms of presence and absence that in 80% of the cases the data agree. But you see in the graph there is differences in agreement between the type of sites like lake, spring, stream or wetland. And so what explains these? So there is more disagreement at the lake sites. These are very large sites. There's also more disagreement when abundance is low, so it's more difficult to find the snails. There's also more disagreement with the higher species diversity, so when there are many different snail species in the site. If you compare the data again, we see that there is the same trend in relative snail abundance, but consistently the citizen scientists report lower abundances compared to the experts. So the dotted line is each time the citizen science data per snail genus. Then the second part where they act as communicators, here Maxon Agnolito will talk more in the next session. And we also have PhD students in ATRAP who are doing sociological studies, anthropological studies, in order to find out which knowledge gaps are there, which myths and taboos regarding schistosomyces. And they use this information to co-create contextualized communication tools that they will use in the community-led awareness campaigns. And finally, we also created encounters between the citizen scientists and the policymakers to co-design policy recommendations. So to conclude, we think that citizen science can simultaneously increase monitoring capacity and public engagement in vector-borne disease control, because it can really generate unprecedented data sets in terms of spatiotemporal resolution. And at the moment, for example, we have more than 35,000 snails collected by the citizens in Uganda alone. The use of Kobo Toolbox app and artificial intelligence, which I didn't talk about now, given the time constraints, but these facilitate upscaling of citizen science projects and implementing them in remote areas. We also found out that the citizens are really trusted, or the citizen scientists are trusted by the community, so they can really act as a bridge between the scientists and the communities, so that allows a two-way exchange of preventive measures on the one hand and community needs on the other hand. So citizen science can create ownership and lead to sustainable practices if there is continued communication and engagement between all parties. This is really crucial, we found out. So thank you for your attention, and questions will be for later.
Thank you very, very much to both Mercy and Tina there. A fantastic presentation. I'm just trying to stop your slides. There we go. Thank you very much for that. We were blown away when we first heard about this a few months ago in the first session. And I think a lot of people maybe are seeing this for the first time, but it's a fantastic bridge between the citizen and the policymaker as well. One of the slides you were showing, all of that data collected, all the sampling done, all of it has led to that kind of the community sitting in the same room as the policymaker. I think that's a fundamental endpoint, something that needs to be talked about a lot. I'll be interested to know about how the AI would be used to upscale. I think that'll come into the questions at the end, but thank you very much, both of you. So Marco, we're going to hand the floor over to you, Marco, for your presentation.
Yeah, it's fine for me.
Brilliant. So then we'll hand over to you, Marco. Thank you for that, and I'll back out.
So good morning, everybody. Thanks a lot, Cameron, for inviting me to this festival. I'm very happy to be here and to share with you some results of our work we are doing to fight Asian tiger mosquito on a small island in Italy, in Mediterranean Sea. I am Marco Salvemini. I am professor of genetics at the University of Naples. And since some years, I'm coordinating a project of citizen science project on this small island to try to give a contribution in the direction of the development of novel tools to involve communities in participating in this kind of control campaigns, monitoring and then control campaigns. So some few words about our island. Procida is a very small island in southern Italy, in Mediterranean Sea, and it has some unique features for a mosquito control and monitoring and control program that are a very small size, only 3.7 square kilometers. There are a lot of tiger mosquitoes there, and also a high density of humans. And also we have an administration, so politicians and citizens that are quite sensitive to environmental issues. And so with these points as premise, we started some years ago a project to collect baseline data about the mosquito population on the island, and then to arrive to a control phase using sterile insect technique, so releasing sterile males on the island. So we started in 2015, and we did a lot of activities with the citizen. With their help, we collected the baseline data about the temporal and spatial dynamics of Aedes albopictus on the island. We tried to involve people also doing public activities on the island, not only the scientific one. We performed 3 years ago a first release of sterile males to test the flying capability and survival and dispersion rate of mosquito on the island. In this picture, you can see some volunteers, some students from our university, and this guy, close on my right, that is the mayor of the island that participated with us as volunteers. So we tried also to involve politicians directly as a citizen scientist, not only to talk and discuss with them for permission on other legal issues. So we recently published the results of our work on close-neglected tropical disease. So if you are interested in this study and you want to learn more, you can have a look to our paper. So during these years, we did a lot of effort, as I said, to engage the community and to invite them to participate in our programs, organizing public events, for example, to teach citizens something more about mosquito biology and control methods, and about good practice they could apply in their properties to limit mosquito spreading. We had a special focus on students and young people, because they could be trained and to become expert adults in terms of mosquito managing in their properties. And despite the success of many of the activities we performed on the island, as scientists, we experienced that we have not enough and proper skills, maybe, to foster engagement with the community. Community engagement, in our experience, was not only a matter of study and explain to people, and the community itself is more than the sum of the individuals it is composed of. So we understood that relationship building with the community is the most important part of a program, to apply then a control program on the island. And we decided to focus on these aspects, interrogating about how to perform a proper community engagement. So we tried to imagine an innovative way, trying to involve, and also a transdisciplinary way, involving artists. a special kind of artists that we have in Naples at Fine Art Academy. They are relational artists. So they are artists that use relationship building as an instrument to create artistical installation or events. And these teams of students, there are more than 20 students working together, are coordinated by Prof. Franz Jandolo at the Fine Art Academy in Naples. So the idea we had is to involve them, and with their help, trying to design a community engagement campaign to be held on the island this year in 2022, with the idea to transform a classical mosquito monitoring and control campaign into a mosquito, let's say, cultural event. And to do this, we try to add to the classical positive outcome that could be obtained by a mosquito control campaign, such as the mosquito density reduction. We try to add something in terms of outcomes that could be of interest of a community. For example, we will try to increase community cohesion or increase social inclusion in this kind of project. And also we will try to induce good feelings about taking care, to take care about common good, no? And not only private interests. So now in next slides, I will show you some details about what we planned. And our project, the project that we designed together with these artists, has a name that in Italian is non io ma noi, that means not me but us. And this name contains all the idea of our collaborative approach. So the project is organized in 4 phases, and each phase has a special declination of the title phases. For example, phase 1 is not tree but forest, just to again reinforce the same concept of collaborative approach that we want to do on the island. And phase 1 is focused about gift. The gift is the starting point to create a relationship with people. So we designed a gift that is a bar of chocolate with the map of the island on. We designed also the packaging of this product, and this product will be presented to people on the island to start interacting with them and explain our project and our objective and trying to involve them. And also this product will be produced on the island by local pastries, and then the whole project will be presented to them at the end of the project. They can use this for free to product as a souvenir, for example, hoping to have also a small microeconomics impact on the economy of the island. In this short video, I will show you how these artists will have an impact on the island and how their approach is based.
Abbracciarti ancora con la mia mano sola, posso toccarti ancora, so che puoi capirmi, anche senza parole, da noi non è servita, e mai ci servirà, tra un po' sarà il momento di tagliarmi ancora, che dolce sensazione proverò.
I hope you can hear also the audio, the sound of this video. So during phase 1, we will also ask people to be part of pictures by which we will produce an artistic representation of the community that is participating to our project. So creating a way to show the whole community participating to the project in one shot, using this let's say, murales. Then in phase 2, we will focus on entomological actions. So we will perform a second round of door-to-door visit to private properties on the island, using the relationship we were able to build in phase 1. In each property, we perform together with people the elimination of as much as possible larval breeding sites, and then we will provide people gravity traps. We will provide them special traps to capture adult mosquitoes. So that there will be mixed teams with artists, volunteers, and scientists together doing these actions in, let's say, 500 houses on the island. And the idea is to place 1,000 gravity traps on the whole island. We are also working on the packaging of these gravity traps that should contain also the instructions on how to use them properly and some details about mosquito biology. And then we will ask to people to send us pictures of the sticky trap that are located inside the gravity traps. And these images will be collected using an ad hoc smart app that we are developing. And then we will apply artificial intelligence to count mosquitoes, to perform mosquito identification and to count the numbers. And all this data collected by people will be used to create a video mapping, so a digital visualization of the results of the project in real time, week by week. In this demo, we represented people participating and collaborating as connecting dots moving on the island, and then we will add on this island map of the island the number of mosquitoes captured during the weeks. Then we have a phase. In this phase, in phase 2, as done in phase 1 with pictures, we will ask people to perform a 3D scanning of themselves, and we will use these models to create a second artistic representation of the community that is participating in our project. And in this short video, I will show you the concept of this idea. So in phase 3, instead, using the relationship again built up in previous month, a public event in the form of a fest will be done on the island. These artists call this event an anthropological relational sculpture. And I will give you an idea of what it will be in this third short video.
Questa è la storia di Cicerenella, la donna più brava, la donna più bella, è napoletana ed è nostra sorella, e questa canzone è la sua tarantella.
Colori di Napoli, confini spagnoli, ne vanno bravo dopo.
Cirinelle tenevano, cantane, Cirinelle tenevano, cantane, Cirinelle tenevano, alle tutte le notti va a cavallo, e se incievo passaggio la sella, chi le guarda, Cirinelle, e Cirinella, si buona e bella, e Cirinella, si buona e bella, e Cirinella, si buona e bella, e Cirinella, si buona e bella. Hey! cicerelle tenevano un culo che appare con un funatura e lo mostrava la coppia di stelle, chi le cu, cicernella, che cicernella si pone bella, che cicernella si pone bella, che cicernella si pone bella, che cicernella si pone bella. Hey! Che cicernella mi si pone bella, che cicernella mi si pone bella, che cicernella mi si pone bella, che cicernella mi si pone bella.
Cuore di Napoli, vivi i ragazzi che l'hanno organizzata, vivi il cuore di Napoli.
So in this video you saw an action organized some years ago by these artists in Naples that is called the Heart of Naples, and we will try to organize a similar things but focused on the mosquito control team, and also during this event we will present the 3D installation representing the community and the video mapping of the results produced during the project in real time. Then we will have a final phase number 4, that we will do in September of this year. And in this phase, we will try to evaluate the effects of our action on the territory, not only by an entomological point of view, but also trying to analyze the socio-economical aspects of the action we will perform on the island. In 2022, Procida will be the Italian capital of culture. So this is a very special year to try to show, to try to organize this kind of approach to involve people in a mosquito control and monitoring program. And our project is inserted in the official program of this special year. It is one of the 44 cultural projects that will be performed on the island, and its title is Open Science for a Democracy of Knowledge. And so we really hope to be successful in engaging the community and to have in next year's good results to show you, to tell you. And at the same time, we hope that our approach could inspire other similar actions to fight mosquito together all around the globe. So that's all. Thank you very much. These are all the students of my department and in Fine Art Academy that are participating. This is the day 0, 1 month ago, when we started the actions on the island. Thanks a lot.
Thank you very, very much. Grazie mille, Marco, for a very fun, I mean, people are already commenting on the chat, Wangari Wamboy, a big wow, research made fun, simple and involving everybody. Certainly immersive. And I think it starts to answer some of the questions that were raised in the first session actually this morning, in terms of how do you include and keep those communities included, their voices, all of it. I think you've really kind of captured that in an amazing way. And for people who want more information on that, there was a session that we did with Marco a few months ago. It's on our website. I'm sure we'll flash the link up as we go through. But I mean, amazing. It's such a serious topic as well, given the spread of dengue worldwide and the threat that it's posing, not just to the tropical kind of the typical kind of tropical climates, but we've had autochthonous cases in Spain, France, Italy, Portugal, Madeira. So this kind of thing might become a blueprint of how to involve the citizens for these entomological preventative measures that you've mentioned. So amazing. A lot of people are coming through. Professor Rod Dillon from Lancaster. Yes, amazing stuff. Comfort, fantastic presentation. Joanne Butler, thank you for the fantastic work. So it's been very well received. And it was certainly an amazing involvement of the asset of the community itself. So I think there'll be a few questions coming through to the end. Thank you very much for that, Marco. Thank you. That leads us to our next presenter, Professor Matt Todd from University College London School of Pharmacy and the Open Mycetoma Project. And so, Matt, I think we've heard a lot here about communities from a preventative measures kind of perspective. We wanted to add balance to the session by having a look at the curative side. Is there an involvement and engagement that citizen science can bring forward for the preventative side, the curative side of the equation? And so I think for that reason, we wanted definitely to bring you in. We were really blown away by what you're doing in terms of the open mycetoma work. And so I think enough from me and over to you. Matt, do you want us to do you want us to start your presentation on our side?
Yeah, that would be great. Thank you.
Brilliant. So I'll come off the camera for a second.
Oh, great.
Oh, it's there. Brilliant. Yeah.
Fantastic. Okay, great. Yes. So thanks for that intro, because I mean, I guess the difference between the wonderful talks that we just heard and mine is that in my research, we're talking about discovering and developing new medicines. So real, small molecules that of the kind that are normally developed by the pharma industry. And we've been interested for many years now in thinking about ways in which we can develop medicines in areas where the pharmaceutical industry struggles to deliver. And one of those is in neglected tropical diseases, because there's no money to be made in that area. And so the investment is very little. And so what we've been doing is trying to think about, well, if we don't use the pharma industry for this, and if we can do things differently, if no one's ever going to make any money from this, and we need these molecules just for the public good, then that frees us up to do all these kinds of things openly, without any worry about secrecy and patents and stuff like that. So we can use open science to develop new medicines. And mycetoma is an interesting example, because it is a dreadfully neglected fungal infection that is in Sudan and related countries in Africa and elsewhere around the world. It used to be called Madura foot. And I'll talk about that in a minute. Just by way of introduction, I just need to talk about what I mean by open science in its context, where we're talking about people actually making stuff in labs and what have you. So yeah, I have control of the slides, which is great. Okay, so this is just a summary that some of you may have seen before, just to define what I mean by open science, because a lot of people talk about open science as meaning that you can read papers for free. And I really don't mean that. I mean something very different, which is that people, anybody, can participate in the work as full contributors. So you can see what's going on in the project in real time, and as a result, you can take part. You can do something useful. It's a little bit like Wikipedia in that sense. You can see an article, you can edit it if you want to. You know that your contribution is useful because you're up to date with where things are, and you have all these access rights and freedoms to participate. So the idea was to do this with science and lab-based science, where we're trying to develop new molecules that could be medicines. So for many years now, in fact, our first project started 17 years ago, actually, for improving the medicine which is used to treat schistosomiasis. A molecule which many people have taken called praziquantel is given in a great big pill. And we were working with WHO with an open science project to try and improve that pill. And that was the first project we did that finished in about 2010. And since then, we've been doing drug discovery, mainly for malaria, but also for antibiotics and TB, and as I'll mention, mycetoma, using these rules here as a guidance. So all data and ideas are shared essentially in real time, because everything is just put online. anyone can take part and there are no patents, and then there are some other rules there. And what that means is we have lab books and we have public discussions, and all the data is open and can be reused, and social media takes care of a lot of the sort of community building. And the result of doing things like that very openly is that you get spontaneous inputs from everybody. I mean, from student cohorts in universities, through to just people who can see what you're doing, through to senior people in the pharmaceutical industry. I mean, a real range of people have contributed. That's the kind of social conclusion. And so these are the kind of molecules we're talking about. These are the ones that we're looking at for malaria, just to give you a sense of what we're talking about. Starting points in the public domain. We tweak the molecules atom by atom in the lab. We evaluate them. All the data is shared. And we're trying to progress what are known as hit to lead projects, where you start with something which is okay, and you're trying to convert it into something which could go into clinical trials. That's the basic idea. So as an example of how this works, one example which I've spoken about before, but it gives you a sense of what's going on. We in one of the malaria projects, we had these molecules here that we needed, and we couldn't make in our lab. We didn't have the bandwidth. We tried to make one and failed. And so we put this, appeal out to the community for anybody to help. And a student who was just finishing his PhD in Edinburgh. So we were the core team in Sydney in Australia, and this guy was in Edinburgh just finishing his PhD. And he saw that we needed the molecule, so he started making it. And he shared all of the data online so that we could see, the quality of the work. And he finished a bunch of molecules, including some of the ones shown there, and became an author on the paper that came out. And it was a completely separate, federated project for him. And we could trust him because we could see all the data. And this has happened lots of times over the course of some of these projects. He's free to act. He knows his molecules are necessary. And we know the work is of good quality because all of the data is shared. So it's quite simple structure, really. And over the years, this has been the core of some really fun crowdsourcing projects with student cohorts in universities. So rather than a student cohort making the same molecule as last year, the idea is you make new molecules as part of real live research programs. And it brings us up, some challenges associated with doing that, because, if no one's made the molecules before, then you can't criticize a student who can't make it, right? Because maybe you can't make the molecule. But it's fun, and I think it connects young students in universities. Many of these were first-year undergraduates to real live research, because the molecules are evaluated, and they go into papers and stuff like that. One example where we used a project with school students, as 17-year-olds in Sydney, was where we worked with a cohort of students and their very talented teacher in the school's lab to make a molecule which had been the subject of a big price hike, a drug called Daraprim, which the rights to which have been bought by a company called Turing Pharmaceuticals, and the chair of that company, Martin Shkreli, raised the price of the medicine by 5,000% overnight. So we worked with a school to make a sample of it as an educational exercise, but it caught the attention of the press because the little vial that student is holding in that picture is worth $150,000. So it became a project about, well, how can medicines be so expensive? But ultimately, it was a fun thing to do on our platform because the students could work and share the data, and we could see the quality of the work was very high, and they could get advice from people. So it was a fun project. The kind of things that we do in the lab with undergraduates is that we do fairly complicated organic syntheses. So these molecules are not trivial to make in many cases, and in some cases, so in the purple text there, things are provided by postgraduate demonstrators. But the orange things there are where the students step in and work in teams to progress the chemistry. And it's a really fun thing to do because the postgraduate demonstrators can talk to the junior students and impart wisdom and experience. And yet at the end, you do have an actual molecule, which is then sent off for evaluation. So we cloned all this stuff for a project with DNDi in Geneva. They and Eisai, the company, are involved in a clinical trial for a medicine to treat mycetoma, this nasty subcutaneous fungal infection, which is very debilitating and for which there are currently no cures at all. And so we wanted to try and act as the starting point for new medicines, but beside the clinical trial, which is ongoing, we wanted to try and think about, well, what other medicines could we think about? that could be developed for mycetoma. So we started this project, and we've been working on molecules, again, like this. This is one of the series of molecules we're working on, and which we published a few years ago now. Pretty simple structures, not the best drugs, I think, but pretty simple structures which we're trying to optimize still. And working with students around the world on this. And we're making molecules. We're also trying to use those molecules to think of better ways of treating the disease. And so we're measuring things like how well the medicines penetrate the fungal's defenses. Some of these neglected diseases are really nasty and difficult to kill. In this case, the fungus wraps itself in a kind of protein coat in vivo, and the medicines that work well in the lab don't work well in vivo because they can't get in, and we're trying to solve that problem too. So it's a bunch of molecule synthesis, but also trying to think about how to improve the delivery. And so this is a project where we got some money from the Royal Society to run a partnership grant with a local school in Kent in the southeast of England, which allowed the students to make molecules for the project also. And the amount of time that they have available is pretty small, so often these projects rely on the commitment of teachers, which they certainly have at 7Oaks. And the pandemic really messed up our progress here, because the schools had to shut down like everyone else, and our lab in UCL had to shut down. So the progress we've made has been much less than we had hoped, but the students have been doing experiments and trying to make molecules. And you can see what they're doing, because one of the key bits of the platform is that we have an electronic lab notebook, which is open to anybody. That's one of the key bits of technology. So you can find things with Google and stuff. And so, when the students do work, they can share exactly what they're doing. And here they're trying to cool something down to crystallize it. So it's a really nice way of staying in touch. The school's only a few miles away, but, we're not going to go there on a regular basis. And when they have molecules for us to analyze, we can just get the molecules up into UCL and use expensive equipment, which we have at the university, which the school doesn't have. We've got multiple series of molecules here. The reason I'm highlighting this is because the phenaramols, the first series, are quite hard to make, particularly in a school setting. Some things are a bit unpleasant. And so we are probably going to shift to this series 2, the aminothiazoles, which are a lot simpler to make. And again, we've kind of validated a synthetic way of doing it in a university. And now the school can step in and figure out which molecules they can make that are new to the planet. So, new stuff they can make. And I think the chemistry is a bit more suitable for a school setting. So we're excited to try and do that next, I think, to transfer to something which is a bit simpler. Just in the last few weeks, we've had multiple new contributions, again, spontaneous contributions from the community, which is really exciting to see. On the left there, we've had some natural extracts that look promising on related pathogens that are coming in from a team in Kenya. And on the right there, a medicinal chemist with lots of experience in Germany saw what we were doing and noticed that he had some molecules that are a little bit similar to the ones that we have, but are kind of merged with an anti-malarial medicine. And he's sent us a bunch of samples, many samples to evaluate there too. So, from a sort of from a small core of people who are running the project, we tend to get contributions from the outside coming in spontaneously, which is, a really wonderful thing to do. As a PI, to have this kind of, involvement from people spontaneously is just fantastic. And I guess that's one of the things here, which I'll describe very briefly, is that often a scientific project can be quite a closed team, and you define who you want to work with in advance. But open projects can mean that you have direct interactions between students, which again we see all the time, without crusty old professors like me getting in the way. And also you have people who are not part of your original project team able to step in and contribute, which is where some of the big value comes, I think, is that people can see what's needed and step in and contribute, knowing that their contributions are worthwhile. So there's a few things that are needed, in terms of how other people can get involved. We always need more chemistry. That is often the rate limiting step. We also need people to sort of examine the structure activity relationships of what we've got. What trends can we see? What should we make next? We need help with things like paper writing if you're not a lab-based person. And also, we need people who are wanting to be sort of, project managers. Lots of things go on in these projects, and we have regular meetings, and we have to write up minutes, and we have to assign actions, and we have to chase people for things, and project management like that is also really always needed, I guess. We've also had to get a bit creative about advertising. So On the right there is a picture of the windows of my lab in London. They're on the ground floor of the School of Pharmacy. So if you sort of stand on tiptoes, you can see in the lab and see the hoods. And so, we get people passing all the time because it's a busy part of London. And so in the window, we've put these big posters that mention the Mycetoma project. And there's a huge, great QR code there, the blocky thing. And so if you're standing on the street and you use your phone, you can actually use that to get a web link, which takes you to the web page for the project. So I don't know how many people we've got into the project, from passing pedestrians on the street, but it's worth it because, it's nice to try and involve people in what's actually going on in the lab today, if we can. So just some thanks. There's a snapshot of the last meeting that we had I think a couple of months ago, where some of the project teams came. Huge amounts of thanks to Wendy van der Sande, who's the biologist in Rotterdam in the Netherlands, who does a lot of the biology and is like the world expert on Mycetoma. And then a bunch of other chemists in Sydney and in London, and contributions from New York City and the school at Sevenoaks and other people who've started to contribute more recently. Hopefully together, we'll find something good that will act as a starting point for real drug development for this horrible NTD. All right. Well, thank you very much for your attention, and happy to answer any questions.
Thank you very, very much, Matt, and all the wonderful people at Sevenoaks as well. Still can't believe that it's a high school project. I mean, that's absolutely amazing that their work will ultimately be used into producing, we hope, a new therapeutic for this disease. Absolutely amazing. We talk about citizen science, and there's always this kind of caveat where you're kind of talking about the affected community and engaging on the ground. But this is very much for us a very, very interesting approach to kind of obviously the very complex side of the curative side. So I think that's an amazing project, and I'm assuming anyone who's in the participants, anybody wants to get in touch with you, Matt, can either use that QR code or just get in touch with you in terms of some of the issues that you said you need help on, the copywriting and that kind of stuff. So hopefully some of the participants, you're getting some questions coming through for that as well. So, I mean, thank you very, very much for that. We've been rejoined by Mags, Dr. Mags Leighton from the University of Durham. Hi there, Mags.
Good morning.
I think we can. Good morning. There. Actually, I see it's playing now. So I think I'll just hand over the floor. I was just going to quickly introduce you. We talked about. Well, we had Matt and yourself in to balance the panel because obviously we wanted to see how science communication can kind of impact the curative side of things and to kind of demystify very complex kind of the biochemistry behind certain diseases, these diseases in particular. And this project on cutaneous leishmaniasis that you've put forward at the University of Durham, I think is very, very interesting. And I think I'll just hand over the floor. Thanks, Mags. All right. Thank you.
Thank you, Cameron. And I'm sorry about I'm wobbling in the picture, I'm aware. So joining by mobile phone, and I'm just needing to hold it up in a place where you have a chance of seeing me and I can see you. So please bear with us with this. Very lovely to be able to join you this morning. Our project, which is a global network for neglected tropical diseases, which is a bit of a mouthful, known as the NTD Network, is a Global Challenges Research Fund supported project via the UK's Medical Research Council, and our priorities are leishmaniasis and Chagas disease. And we have been, since 2018, we've been building an equitable partnership around the world with our partners in South America and India and Pakistan, as well as the UK, looking for new solutions for two trypanosome-mediated neglected diseases, and these are leishmaniasis and Chagas disease. And it's in particular that I want to speak to you today about cutaneous leishmaniasis. So of the leishmaniases, we have over 20 species, of which Kala-Azar is the one that tends to get most priority. Visceral leishmaniasis caused by Leishmania donovani and infantum. This is a neglected disease which is lethal if untreated, but there are other forms of the disease. Now, in particular, cutaneous leishmaniasis isn't prioritized so I would say it's the most neglected of all of the neglected diseases, because it's not technically lethal, but it's absolutely a destroyer of lives. So the gift of being able to partner with our colleagues in South America and in Pakistan and in India is that it's been able to put us in the UK in touch with people for whom these diseases are a reality. So you can see from the WHO map here that we have cutaneous leishmaniasis in our areas where we have colleagues, and also in areas across the northwest of Africa and the Middle East. And it was in recognizing that not just these diseases, but the people that work on them are actually a neglected species. Part of our program has been to educate early career researchers in some of the very scarce and effective laboratory skills that can be used to screen for new solutions, drug products and natural products. It was this very endeavour that took us to Karachi in November 2019, and our picture here is of our wonderful students, which we were able to bring together from across the Middle East. So a whole range of endemic countries in which cutaneous leishmaniasis is endemic and a huge problem. And I'd just like to highlight for, particularly if we have some listeners from any of these areas, you are so needed. These wonderful students, colleagues from all over the Middle East gathered with us on this occasion, and they bring not only their very direct experience of the disease, but also they come from countries with huge ecological diversity and a great wealth of a long history of natural products use. And really, kind of these people and the rest of you listening, you are our hope, and we really need going forwards to be able to bring you on board with us and to tackle this globally. But for me, this getting onto the creative part as a part of being able to visit our colleagues in Karachi. In the UK, of course, we're not in an endemic area, and whilst my team have been involved in research into leishmaniasis throughout their full careers, we haven't actually ever had the experience of meeting patients. And it was this visit to Pakistan which it just made everything become so real for me. So you can see in the picture here at Jinnah Hospital in Karachi, the wonderful Dr. Bahram Koso and his clinical team. These are some of the most amazing doctors I've ever met. And we had a chance to meet them. And in particular, they introduced us to some of their patients. So they have, in Pakistan, they have a seasonal rise in cutaneous leishmaniasis cases. We can see just now that this is the typical situation for cutaneous leishmaniasis through the year. We are just coming into the season where the epidemic is at its highest. And I can say that our colleagues are telling me that due to the results of their nation's clinical resources being diverted, of course, to COVID, is that the cutaneous leishmaniasis situation this year is more acute than ever, and it's really overwhelming for them. So the people that are suffering from this experience end up with these horrendous open sores on their faces. These images are actually from Dr. Yasmina Khan, who's one of the clinical team you just saw. And the only treatment that they have available is injection of pentavalent antimonials directly through the top crust of these sores. The reason that this is so difficult to treat is that the current treatments that we have are just unbearable for the patients. So what moved me deeply was a particular family that they introduced us to who were in the clinic that day. So these are images of the treated scars from 4 children from one family. Now they were there. They were all under the age of 10, and they were there with their father, who was a labourer working for a day wage in the city of Karachi. He and his family visited their grandparents near the Afghan border for all of 10 days, and all of his children, when they came back, they were all infected with cutaneous leishmaniasis. And whilst it's a disease spread by biting insects, so the sores themselves are not infectious, we were horrified to hear that these poor children were denied access to their school by their teachers because of fear of infection risk. And the huge consequences of this for people's lives really came home to us on this day, because it's not only are these children at this young age being denied proper access to their education, but the culture that they live in will mean that these scars will follow them all of their lives. Also, in order to get them to hospital for frequent treatments, their father had to give up his day's wage. Now, for people, this raises a huge issue for people in endemic areas who are living in poverty, is that in many instances, and these children were lucky because they were able to easily access treatment, but in many instances, even a treatment which is free is too expensive for people who are living day by day and need the wages. So this situation really brought home to me just how vital it is that we find ways forwards and that we find solutions to these diseases. The next slide was a plea from the doctors themselves. Here's Dr. Yasmina Khan, whose images you just saw. What we really need are drugs that don't have to be injected through these sores, that can be delivered as a tablet form, that the patients can endure, and crucially, that developing countries can afford to a national programme. So if we just come out of the slides, Cameron, and I think the safest thing to do is if you then play the video, I would just like to show you what our response managed to be. So I'm just happy to be able to say that whilst we can't do anything particularly quickly about the need for new treatments on account of the fact that drug development is you need a vision of at least 20 years to be able to get through the many processes of the drug pipeline. The work that we have begun on our project, we're aware that other people will finish. But what we have been able to do is really, in my role, which is a project manager, I'm also responsible for communications on the project. And I had an opportunity to apply for some money internally at Durham from the Matariki network, which was for outreach. And I was inspired to try and do something with this harrowing tale of this family. The images that you saw on the screen there showed a range of responses to the treatment, and this is typical of the complexity of cutaneous leishmaniasis. We have everything from lesions that will heal quite quickly to ones which won't, and that reflects the different species of leishmaniasis which are behind the infection. But we realised, speaking to our colleagues in Karachi, that whilst there's many videos available for explaining the life cycle of leishmaniasis and explaining some of the biology, there wasn't anything really that touches on this side of the patient's experience, and that is able to show really the patient's story and why the the fears around infection risk are misplaced. Cameron, is the video able to play there, please?
Yeah, we'll play it now. We'll play it now, Max.
Thank you.
Did you know that poverty and risks from many neglected tropical diseases are interlinked? Many of these diseases can be difficult to recognise, yet they often cause serious societal and community problems. Let's look at the example of cutaneous leishmaniasis. Salima and her family live in Karachi. They have just returned home from visiting their grandparents in rural Pakistan, where deforestation and poor sanitation have created ideal conditions for sandflies. These tiny biting insects carry several types of leishmania parasites. Salima and her brothers became infected whilst they were playing outside. Skin-based leishmaniasis causes painful, crusted, open sores. The children's father decides to take them to Jinnah Hospital. Treatment is painful, and around a third of the infections don't respond to the drug at all. Because the children's father loses a day's wage each time he takes his children into hospital for their treatment, the family has to cut back on food in order to meet the rent, and often go hungry. This is not an uncommon story. There are more than 1 million new patients with cutaneous leishmaniasis each year, and around 40 million survivors carrying scars, causing serious physical and mental health problems. Ignorance surrounding this neglected tropical disease often causes sufferers to be excluded from education or employment opportunities. In some cultures, women and girls with these scars are considered unmarriageable or even forcibly separated from their children. After many painful weeks of treatment, Salima's brother's sores have healed but have left unsightly scars. Salima is not as fortunate, and her terrible sores remain. She is reluctant to go outside and believes her life is over. Diseases of poverty can be overcome, but there is much work to be done. We are working towards better understanding, more patient-centred care, and more consideration for the families affected. Yet, there is an urgent need for more research into better diagnostics, treatments, and vaccines to bring new hope for people like Salima. To find out more, visit the links on screen.
Thank you very, very much for that.
Great.
Thanks a lot for that presentation, Martin, and thanks to everybody in this session for the wonderful presentations. We've kind of reached the end of the time. However, I do think it's fair if we ask a few questions. So would it be okay just to prolong it for another 10 minutes, just to get a few questions in from the audience? Are you all okay with that?
Yes.
Yeah, okay, thank you. Thanks lots. Well, I had some questions in mind, but I think it's just fair to go to the audience questions. And I'm just in no particular order. There's a question from Edith Nwanko. Very interesting presentation regarding Procedure Island. Community engagement very necessary for achieving control. She has a concern about the huge cultural activities involved just to drive the message home. So her question is, how did you get the funds and resources for sponsoring such cultural activities? That's to Marco, but I'm going to open that up as well in a different way to everybody else. But I'll let Marco answer that firstly.
Okay.
So how did you get the funding, resources?
Yeah. So our project is not just to let the message go home. No, it's focused on community engagement. So our idea is trying to add the cultural events, but to let people participating. So in the central part of our project, we are asking people to do citizen science activities with us. About the funding, let's say that the most expensive part of the project is to have 100 students on the island for 40 days. But also in this case is a matter of community engagement, because we are asking people on the island to host for some days these students in their houses. So instead to look at a simple solution, just booking hotel and let people stay in there, we are trying to put people together directly also during the hospitality part of the story. No? So this is one of the solutions. So we solved this problem of resources required, just trying to decrease them, the level of resources required, involving people also in these aspects of the story. Then my university and the Fine Art Academy are putting money in this story. They are investing some resources, and we got other funding from the cultural program that is this year is especially to do things because there are some money coming back from our regional government for the cultural activity on the island, and we get some funding also from this side. But however, we are trying to do something that should be sustainable, so trying to decrease the cost, involving people in all the aspects, also in some logistic aspects like hospitality, for example. This is the way we are
doing. Great answer. Thank you very much, Marco, for that. Thank you very much for that. I'm just aware of the time. I just wanted to kind of open up a little bit and just ask. I know, Aiden, you've answered a couple of questions. There was a question from Dr. Wangari Wambui from KU Leuven, asking in terms of the web link for the digitized collection, which you've sent. There's also a question from Dr. Sajida Riaz from Pakistan, who's working on schistose snail hosts and has samples, and it's kind of a direction of research she wants to progress in. Is she able to contact yourselves and Tina and Mercy and Aiden in terms of that?
Of course. Yeah.
You can't speak, say that's fine on the spot. What I wanted to ask you really was that there's a specific question there for also for Professor Matt Todd from Alan Carlin Wu from Ireland, asking how to scale up the production of new drug molecules after they were synthesized in the school lab. Will patents be applied? So specifically to you, Matt.
So everything we do is no patents. That's one of the key things. So there's no exclusivity on the molecule, so that they can be made by anybody. That realistically doesn't mean that everyone's going to start making them and selling them, because of course to make and sell something, there's a huge amount of oversight involved in that process. But I mean, the examples I know of are things like NGOs and others forming manufacturing agreements with companies. So the new and improved pediatric praziquantel for schistosomiasis that's recently completed a clinical trial in the Ivory Coast, that is being made under contract from Merck by a company, I think, in Kenya. So often you find these big scale arrangements where the molecule is made on a large scale, where you don't have traditional patent agreements. So yeah, any molecule that is made by a team goes through clinical trials and is approved as a medicine. You would then have to negotiate manufacturing. But given that we have a generics industry, which is really efficient these days, these things are not terribly difficult to sort out.
Okay, brilliant. I wanted to kind of ask a wide question. I was trying to pool a lot of questions together. Aiden, in your initial slides, you had this slide where you're showing the visitor attraction, the public engagement background of the museum, the archiving of samples, research, education, some of the kind of areas that a museum is involved in. And from hearing all of these other presentations, is this some of the gaps that we're talking about in terms of training, collection, sample collection, the marshalling of the public? Are there partnerships, do you think, that could be formed across some of the panelists here? Can some of the museums take up some of this work in terms of getting schools together? You put this QSR code out in terms of trying to attract more attention to the Open Mycetoma project, where you're working with schools. Do museums have a role to play in the future for this type of community engagement, whether it's Marco's engagement, whether it's the University of Durham Mags's engagement, whether it's Professor Matt Todd's type of engagement? It's a question to both Tina, Mercy, and to Aiden.
Shall I start? The public engagement, of course, is a huge part of what we do, and citizen science also is. We've got a number of citizen science projects at the museum. Always the issue for us as a big sort of general purpose natural history museum is engaging with the engagement and directing our priorities, coinciding our priorities with theirs. So at the moment, the focus for the museum seems to be to concentrate, be a little, it's gone a little bit introspective, I think, and we're pushing things at quite a local level, which makes things a little bit more difficult for me. But that's not to say that the reach that we've got because of the size of it, if we can find a way to get in there and harness that, then I think it could be a very powerful thing indeed. Of course, we already, we train scientists from all over the place. We engage at a scientific level very effectively. Broadening and of course also digitizing the collection is democratizing in that it gives people access from all over the place as well. So I think these are all very positive things, and we need to find a way now to capitalize on that and to get that interest.
Oh, that's a great answer. I wanted to ask the same question to both Tina and Mercy, because a lot of the threads that Marco's spoken about, Matt's spoken about, Mags has spoken about, it kind of feels that there's a role for the museum to push this type of engagement further. What are your views on that, considering what you're doing right now? And that's to Tina and to Mercy.
Yes, so our museum, the Africa Museum here in Belgium, is also engaging in citizen science projects. But what we found out, you need, of course, two things: that your collection is being open, accessible, so digitized, and so that's ongoing. At the moment, all the holotypes and paratypes of specimens are digitized, but we need to increase that. That also asks money and personnel. But then on the other hand, you need the citizens. And so the first citizen science projects, we could engage with citizens and involve them in the project. But to keep them motivated and involved, you need to organize some activities. You need to provide exchange moments to keep them up to date, what we do with the data. And so it asks also a lot of involvement from the scientists, who also at the same time need to write scientific papers, as you know. So that's why I think it would be good to have some special funding for citizen science projects within our museum. And we're also looking for that with different museums in Belgium to coordinate there, because these are transferable skills, and it's the same kind of outreach that you need. So it's not a quick fix, the citizen science activities. You really need to put a lot of time in it, or it will not really be successful. And of course, ideally, you will generate scientific data that is really useful for publication. At the same time, increase awareness and maybe even induce informal learning. And if you want to tick both boxes, you really need to prepare well, and you also need a big team which has all this experience. So we're really looking in, and there's some Flemish funding, but not funding on the national level. But now the EU is really putting a lot of money into that. They really believe in the citizen science approach. So big teams are forming to monitor air pollution, but of course we're working on tropical diseases. So we're working in Africa, and that really is very different to perform citizen science in the global south compared to the global north. So we have other challenges. So yeah, that's our perspective. If you want to add.
Great, thank you for that, Tina. Mercy, do you want to add anything to that?
No, I think
there was a question from Aiden. Oh, wait, no. Yeah, after you, Mercy. Sorry, I thought you. No.
No. I actually say Tina said everything really well, and I'd like to emphasize her last statement. Can we copy paste citizen science practices from the global north to the global south? Because it's different contexts, different people, different cultures. So this is a question that is going on even with us here at the museum. But whenever we interact with people, we like to pose this question so that people can think on it and kind of stimulate some debates going on there. Yeah.
This cultural inertia that you mentioned, Mercy, it's a huge point, I think, because there's no such thing as a blueprint, I don't think, for citizen science, but there are some very interesting patterns being formed and some interesting frameworks being put out. Edith has a specific question for Mags, which I'm going to ask. In terms of cutaneous leishmaniasis, great presentation. How do you engage patients in the area of control and protection against the insect vector, the sandfly, transmitting the disease? How would they protect themselves in the environment against the insect, knowing that playing around the environment is inevitable for children? So she's asking really, Mags, a question there in terms of your outreach to the public to make them more aware in terms of preventative measures. Mags, I don't know if you're there.
Oh, hello. Yes. Oh goodness, that's such a good question, and I'm afraid I'm quite outside of my area of expertise to be able to answer that, unfortunately. Yes, the areas where there's highest risk are areas where many diseases are also have an enhanced risk. So areas where there's deforestation, where there's few resources of sanitation and other kinds of infrastructure, these things are compounded. And unfortunately, I don't know of any programs, unlike malaria, where you can target the areas where mosquitoes are breeding. Sandflies don't have that kind of easily manageable, well, not easily manageable, but the habitat for sandflies is really everywhere. So public health measures are, I think, a real challenge for trying to approach it from the point of view of the insect.
Yeah, that's very interesting. I think there's a kind of a question from Bjorn Garing. Thank you for that, Mags. I think, and this question we'll ask to everybody, and we'll have to end it after this. I mean, I have to end the session because the next session is starting in about 10 minutes from now. So the question from Bjorn Garing from Sudan is that to all the presenters, we know for science to be influential, it has to come down from its ivory academic tower and be engaged with the people. What are your tips for organizations to activate people-centered science with the people and for the people? Thank you. This is Bjorn Garing from South Sudan. So he's asking, so I want to ask all the panelists, what would be your tips from your point of view to engage with those communities? What did you do? Was it the tech that you used? Was it the kind of synthesization that you did? The outreach programs? What was it? What would be your kind of a tip? And we'll start with Mercy, because you're at the top right, and we'll work our way around. Mercy, Tina.
Maybe before I say what I have to say, Tina wanted to comment quickly on the previous question. You want to go?
Yeah, I mean, no, just for like leishmaniasis is another arthropod-borne diseases. They have these tiny targets that citizens are using to trap the vectors, which could also maybe be an interesting approach for leishmaniasis control to involve the community. But over to you, Mercy, with your great tips.
No, I actually want to base my tip of my experience from citizen science in the global south. So a lot of citizen science is obviously going on in the global north. What we see is that you need to adapt and contextualize citizen science projects to fit into the context or the area or the location where you are going to be working. A good example is with ATRAP. So our selection procedure, for example, for the participants is different from the typical citizen science procedures or guidelines, as, for example, listed on the EXA website. You look at our facilitation for the participants. We realize that they have to incur transport costs to go and collect data. And for this, we kind of find a balance to kind of pay it back or compensate. It's not a salary, but it's more like compensating for the costs they incur while collecting data. And so we kind of take care of things which normally under citizen science projects elsewhere might not need to be catered for. But yeah, so bottom line is contextualized, adapted to the situation. Yeah. Tina.
That's a fantastic answer. Thank you.
Well, maybe for the Belgian context, I think what also is really helpful to involve schools, because kids, they're really curious. It's easy to engage them, and you might have that trickle down effect, or up towards their parents, that they talk about what they learned in school. Yeah. And so inviting the kids over here in the museum as well. So that's also, and we're also thinking of involving kids in Uganda and Congo, where we already are doing that in Congo with schools.
Fantastic. Thank you for those answers. Marco, to you, you've got a very immersive kind of approach. Is art the weapon? Is art how you're going to do it? How are you going to?
So based on our experience on the island, I think that one tip could be to do things together with citizen scientists, not only to ask them to be involved and produce data for us, but just doing work together in the field. Because the most important part, I think, of the story is the relationship and the trust that you can build with society. But this is only possible if you're doing things together in the field. So in our project, we are participating in producing data. And usually we have half of the work doing by us and the other half by citizen scientists, sometimes in teams together. Another thing we are trying to do is to show people, not directly involved as a citizen scientist, to show people what we are doing in their gardens. So trying to create something like a simulation of the techniques we are using, trying them to experience exactly the same things in a small context. For example, some years ago, we created something like a very huge cage, and we released a sterile maze into this cage, and then we let people enter this cage and trying to capture mosquitoes using electric aspirators. They are looking at doing these things in their gardens, but in that case, they are doing exactly the same. And so this was really an engaging moment. And so slowly a strong relationship with people start to be built on the island. So the idea is not only to ask people to participate, but doing things together. Of course, it's time consuming and resource consuming, but the outcome is really powerful in terms of engagement. So this is our experience.
That's fantastic. And I'm sure you built champions there as well in terms of the community, people you're engaging with, the children, especially talking to their parents and driving that activity forward. So that's a great answer. Thank you for that. Matt, from your perspective, I mean, you know, how are you going to be kind of, what would be your tips to keep engaged with the people? I know you use GitHub, Twitter, and various kind of platforms that the youth are definitely involved in and aware of. Are there any other tips on keeping them engaged and keeping them involved?
Yeah, I think, I mean, I guess, you know, involving others in the research, we do the open science so that people can come along and help if they want to, and that's what it's all about in terms of the advertising and everything. But ultimately, you know, you want to attract people in to contribute if there's a good reason for that interaction to help and to advance things. And you know, so I guess reaching out to people for something, you want to make sure it's a deliverable you need and it's achievable. And that people don't contribute and then don't feel rewarded or that they've helped in some way. So it's important to kind of close the loop and have an outcome or an output or a publication or something which indicates that people have that their contributions have mattered and it hasn't just sort of disappeared into an amorphous project. So that's very important, you know, so that contributions are valued. Trying to show that the research that you're doing is better and improves things is important if you're going to try and raise money for it, which is another key thing because everything's expensive. But I guess I would say that the ivory tower is crumbling a little bit in this sense, because often now when we apply for money for grants, we're asked very detailed questions about what we've done to break down those walls and to involve people. What have we done that is of genuine service to the community? So a way to make the ivory tower listen is to get the funders to change what they're telling you. And if the funders, people like the Wellcome Trust and others, are really interested in how have we engaged the community, then all of the academics will listen.
Yeah, that's a very interesting take on it. Actually, how all of this is influencing funders ultimately. I think that's a very interesting thing. And that's, I guess, why I was kind of saying, is there a lead role for museums to play in all of this marshalling, all of these communities and approach? Aidan, what would be your take on keeping people engaged? That's why I keep putting that into the mix. That is there a natural fit here for museum to lead the charge on these various initiatives.
Well, I'd like to thank you. I'd like to think so, Cameron. I mean, my take on this is that, and I agree with everything that everybody else has said so far. Is kind of when presenting this stuff, to bear in mind the background of the people that you're talking to and how conceptually difficult actually some vector-borne disease with complicated life cycles is for people without a background in biology. And really, if you want to understand how transmission's occurring, how you're likely to pick up these diseases, you really need to get that message across to some of the things that Mags was talking about kids being excluded for schools probably in circumstances where there was no possibility of transmission, all that kind of thing. So for me, it's not just a disease awareness, it's getting the mechanisms across, and I think that's so difficult to gain people's attention almost for long enough. I mean, if you're giving a schisto talk to a non-specialist audience, it can take you nearly 10 minutes just to sort of get the basics across of this complex life cycle and the different ways that it's transmitted. So we need to find ways of doing that, and I don't yet have a complete answer for busy people with different priorities.
Yeah. I mean, that might make, that the animation that Mags showed and some of the work that you guys have done at the NHM might make an interesting exhibition in its own right.
Exactly.
Door opener to these diseases. I think that could be an interesting thing to think. But I'm getting in a lot of trouble if we continue, because we're now kind of quite over for the next session. Some of the comments from the audience, Ikram Guzani, great examples of community engagement, very inspiring. Thank you very, very much, very inspirational. Rod Dillon Marco makes a very good comment. Establishing relationships and trust are key points for powerful engagement. Rod Dillon, again, Professor Rod Dillon, museum folks would love to be involved, but they don't have enough time in the day and week to engage in any detail. Needs funding for folks who work on this specific element. So perhaps all of these efforts combined are going to channel towards a momentum that's going to kind of mean funding changes, or has to change to encompass these kind of citizen science approaches. We look forward to that day. I just like to say thank you very, very much to everybody on this session, and to the wonderful audience as well. I'm going to have to call it a day now, but thank you very, very, very much. Indeed, thanks a lot, everybody. Okay, and just goodbye, just a quick wave goodbye to everybody. Thanks a lot.
Goodbye,
Take care.
thank you very much. Ciao.
Take care.